Hi, my name is Shaira Mallorca Montelibano, STEM 11 Ramirez from Group 4 Gen Biology. We are going to discuss all about enzyme. And for now, we are going to talk about what enzyme is. So, enzymes are the complex protein molecules often called biocatalysts, which are produced by living cells. In specific, enzyme is a type of protein, and all living organisms have enzymes and we naturally produce them. Protein is an important micronutrient that every cell in the body needs. It helps build and repair cells and body tissues, including the skin, hair, muscle, and bone. Protein is also important for blood clotting, immune system responses and hormones, and enzymes. Enzymes are essential for life and play a variety of roles in the body, including aiding in the fight against infection and germs, in digestion and metabolism. Some enzyme aids in the breakdown of big molecules into smaller fragments that are easier for the body to absorb. Other enzymes aid in the joining of two molecules to form a new one. So enzymes are very selective catalysts, meaning they only speed only one type of process at a time. Like for example, when peeling, bruising, or cutting fruits cause them to release enzymes with the presence of oxygen in the surrounding air, then goes into chemical reactions that produce brown pigments through the process of enzymatic browning. Like for example, an apple that turns into brown after a few minutes when it is exposed to air. Thanks.
Hi, my name is Reynaldo Adelcoro Jr. from STEM 11. Again, enzyme are protein, okay? We call them proteins, they are catalysts. These are substances that speed up reaction, and please take note that they are not consumed or destroyed during this reaction. They simply lower the activation energy they need simply catalyze the reaction and then process to catalyze another molecule or catalyze another compound. Okay? So the enzyme essentially can act upon multiple compounds or multiple molecules in a given time. Okay? We can also compute for the rate of the activity of an enzyme. Okay? They are vital to the regulation or metabolic processes and many enzyme protein. So since having many, because there are actually some enzymes which are made up of RNA. So that's sometimes why it's an exemption. Enzyme category and function. Oxidative oxidation or reduction reaction. Transferase, group transfer. Hydrolysis reaction, transfer of functional group to water. Lysis, addition or removal of group to form double bond. Isomerization or intramolecular group transfer. Ligases, joining of two molecules.
Hi guys, this is Gian Rotolano and today we're going to talk about the four components of enzymes. So what are the four components of enzymes? The four components of enzymes are apoenzyme, holoenzyme, cofactors, and coenzyme. First is apoenzyme. So what is apoenzyme? Apoenzyme is an active enzyme. and this occurs of binding organic or inorganic cofactors. These are enzymes that lack the necessary cofactors for proper functioning. Apoenzyme is a protein. Holoenzyme. Holoenzyme are the active forms of apoenzymes. Its DNA polymerase and RNA polymerase are examples of holoenzymes. Cofactor. Cofactor is mostly ions or small organic molecules that are or inorganic and organic chemicals that assist enzymes during catalysis of reactions. Cofactor is a non-protein component, for example, magnesium or zinc. Coenzyme. Coenzyme are non-protein organic molecules that are mostly derivatives of vitamins soluble in water by phosphorylation. Coenzymes are organic cofactors. For examples are NADH and FADH. Hi guys, this is Leandro Toelana from grade 11 STEM Ramirez, and today we're going to talk about enzyme structure and enzyme components. Enzyme structure or protein structure can be presented in 4 ways. Enzymes are made of 21 different amino acids that are linked together through an peptide bond. This results a polypeptide chain, a primary structure. Which order of amino acids is derived from the gene it is transcribed from. In the secondary structure, the amino acids can interact with each other and form a hydrogen bond through a hydrogen atom in the amino group, NH2, and the oxygen atom, COOH, which results in the chain folding in 2 ways. The A helix, where the chain wraps around, or the B sheet, where the chain folds on top of itself. Tertiary structure. The protein could further fold up due to the bonds between the side chains of the various amino acids, forming a 3-dimensional structure. My clue word in this structure is the amino acids could fold up, forming a 3-dimensional structure. And since some of the proteins consist 2 or more polypeptide chains, the quaternary structure is the aggregation of different polypeptide units, and it is the overall protein structure. Now moving on to the topic we are going to discuss next is the enzyme components. So the enzyme components has 4 types, and this is the apoenzyme. These are the protein portion of the enzyme. Its activation occurs binding an organic or inorganic factor. These lack necessary cofactor for proper functioning. Holoenzymes are the active form of enzymes. A cofactor and apoenzyme conjugated together. Coenzymes are chemically different to cofactor, mostly small organic molecules that assist enzymes during catalyst reaction. Cofactors are non-protein component that activates the enzymes. These organic cofactors that mostly derives by vitamins that are soluble by water by phosphorylation.
Hi guys, my name is Francie Cambalaser and I am from grade 11 STEM Ramirez and from Group 4 General Biology. And for today's video, we're going to discuss about the terminologies of our topic or the topic that given to us. So let's start. Let's start from the catalyst. It is a substance that speeds up its chemical reaction without being changed. The next one is the enzymes. It is a biological catalyst and usually a protein. The third one is a substrate. It is the reactant molecule that enzymes work on. The next one is the active site. It is the part of the enzymes where the substrate binds. The next one is the enzyme-substrate complex. It is formed where the substrate molecule collides with the active site of its enzymes. And now let's proceed to the endoenzymes and exoenzymes. What is endoenzymes? Endoenzymes is the enzymes that function within a cell. It is also called intracellular enzymes. And now, what is exoenzyme means? From its own word, exoenzymes is the enzymes that function outside the cell. Both prokaryotic and eukaryotic cell produce exoenzymes. Second to the last is the activation energy. It is a minimum energy required to start a chemical reaction. And for the last one, the transition stage. It is the intermediate stage in the reaction which is the old bond breaks and the new bonds are formed. And that is all for my part. Thank you for watching. Again, my name is Francie Convaliser, grade 11 STEM student from Ramirez, Group for General Biology.
Hi, my name is Eliza Rose Ayabenes from grade 11 STEM Ramirez. Today, I'm going to explain how the enzyme work. So, cells rely on enzymes to kickstart chemical reactions and speed them up, enabling cells to get the most out of the energy resources available to them. Enzymes have a unique way to kickstarting reactions. They work by binding to one or more specific molecules. This is what we call reactant or substrate. Binding occurs at a special region on an enzyme called the active sites. Now, I'm going to explain step by step on how enzymes work using this model. This model is what we call induced fit models. In the speed models, this is when the substrate binds and tightens in the active site of the enzyme after the initial contact. In addition, when the substrate binds in the active site, the shape of active site will adjust for the substrate. Now let's proceed to the process of how enzyme work. First is the substrate will bind in the active site of enzyme. Once the substrate bind in the active site of enzyme, they going to form a what we call enzyme substrate complex. The third process is as the enzyme in substrate begin to react, some of chemical bonds in the substrate begin to weaken, which causing them to link together. The chemical reaction in active site lead the formation of a different molecules, what we call the product. In short, the chemical reaction will proceed where the substrate will converted into the product. And lastly, once the reaction has occurred, the product is released in active site of enzyme, and then the enzyme will return to its original state, and it's free to react again with another set of substrate. And that's how the enzyme work.
Hello everyone, I'm Noel J. Mandala from 11 STEM Ramirez, and I'm here to present factors affecting enzyme activity. There are different factors affecting enzyme activities. The first one is temperature. When temperature increases, the rate of reaction speeds up, and when temperature decreases, the rate of reaction slows down. The second one is pH level. Enzymes need to be at the optimum pH level to work. And when there is a change in pH level, the enzyme activity decreases and the shape will be altered. The next one is substrate concentration. Substrate concentration increases the rate of reaction to a certain point, but an increase in substrate concentration will have no effect when there are no enzyme to bind to. Next is enzyme concentration. Just like substrate concentration, enzyme concentration speeds up the reaction as long as there is substrate to bind to. Higher concentration of enzyme also require greater initial reaction rate. And lastly, there are enzyme inhibitors and inhibition. There are two types of enzyme inhibitors, competitive and non-competitive. Competitive inhibitor is molecularly similar in structure to a substrate that can bind to an enzyme's active site and compete to a substrate that should be at the active site. The non-competitive inhibitor attaches to a other site of the enzyme that is called allosteric site that can alter the shape of active site. The alteration will result in non-optimal position of an enzyme to catalyze a reaction. Feedback inhibition regulates the rate of metabolic pathways when an end product of the pathway accumulates and also stops the starting enzyme in producing materials for the end product to prevent producing more than enough end product.